Okay, so welcome back to this next video in which we are discussing cholesterol metabolism. Okay, right, so we've now had this overview of cholesterol metabolism that is shown here, and what we now want to discuss is how cells such as peripheral cells and hepatocytes can control the amount of cholesterol within them. Okay, so we want to discuss cellular cholesterol homeostasis. Okay, and we've discussed that basically if the cholesterol level goes too low within these cells, what's going to happen is they're going to start synthesizing cholesterol, so both of them are capable of synthesizing cholesterol, and they're also going to start removing cholesterol from the LDL pool. So we've discussed now what the LDL pool basically is. Okay, so in the blood you have these LDL particles, these low-density lipoprotein particles circulating, and we've discussed how these are effectively circulating stores of cholesterol. Okay, so we've discussed that they have this basic lipoprotein structure where you have apolipoproteins in this monolayer of amphipathic lipid molecules, which makes up the outer layer of the lipoprotein, and then inside you have the core. And we've discussed that in the case of LDL, the size of these things is roughly 18 to 25 nanometer diameter, okay? And they have only one major apolipoprotein, which is ApoB100, okay? And key is the fact that in their core you have really only cholesterol esters. That's the main really hydrophobic molecule that you have in the core of the LDL particles. And of course cholesterol esters can be returned to free cholesterol molecules very easily. All you need to do is break that long chain carboxylic acid off, and then you've got cholesterol. Okay, so that is a accessible form of cholesterol there. Okay, and in the amphipathic lipid monolayer here, you have a lot of free cholesterol molecules, more than you would have in other types of lipoprotein compared to the amount of phospholipid that you have here. So these things are stores, basically, of cholesterol molecules. Okay. So, we now want to discuss then the control of cholesterol level inside cells, basically. We want to discuss how do they synthesize cholesterol, firstly. Okay, and we want to discuss the SREBP pathway, which is basically a response to having too little cholesterol. And then we'll go on to discuss what the response to having too much cholesterol is, and that differs between the hepatocytes and the peripheral cells. Okay, right, so for now, let's firstly discuss cholesterol synthesis. Okay, now, cholesterol synthesis, or cholesterol biosynthesis as it's called because it occurs in living cells, is a huge, great biochemical pathway, and we're not going to go through the entire thing. Okay, what we're going to do really is just give the core highlights of cholesterol synthesis, okay? So we're going to look at one of the absolutely key reactions in the cholesterol synthesis pathway, which of course is the famous conversion of HMG Coa into mevalonate. Okay, right, so I'm going to give an overview then of cholesterol synthesis. Okay, right, so cholesterol biosynthesis then, it starts off from the starting material, which is acetyl-CoA. So the starting material for cholesterol biosynthesis is acetyl-CoA. So I'll just draw a molecule of acetyl-CoA. So basically this is an acetic acid molecule, which is a 2-carbon carboxylic acid molecule linked to the thiol group of a CoA molecule via a thioester link. Okay, so I'll just represent the CoA here. Okay, so this box here is representing quite a large molecule called coenzyme A. Okay, which I don't want to draw out the full structure of, so I'm just going to represent it as a box with CoA written inside. Okay, and the only thing we really need to know about coenzyme A is that it has this important thiol group. Okay, so this sulfur here is part of CoA. Okay, and this thiol group has reacted with the acetic acid molecule here to create this link between the coenzyme A and the acetic acid. Okay, and this is an example of a thioester link. Okay, so it's similar to an ester link, but instead of the carboxylic acid group binding to an alcohol group, you've linked the carboxylic acid group to a phyl group here. Okay, now phyl groups have very similar chemical properties to alcohol groups, because effectively an alcohol group is a oxygen atom with a hydrogen atom linked off it. A phyl group is a sulfur atom with a hydrogen atom linked off it. And sulfur and oxygen are actually in the same group of the periodic table, so they have similar chemical properties to one another, and therefore these alcohol and phyl groups have similar chemical properties to one another, hence how they can both react with carboxylic acid groups in much the same way. Okay, right, so our starting material then is this acetyl coenzyme A, okay, which is one of the key products of, for instance, beta oxidation of fat molecules, and also, of course, glycolysis and the link reaction of glucose molecules. Okay, so acetyl-CoA is a molecule that cells have a lot of. Okay, so this is the starting material for synthesizing cholesterol, basically. Okay, and I want to stress again that cholesterol synthesis can occur not only in hepatocytes, but also in peripheral cells. Okay, because both of those types of cells have the necessary enzymes, and of course they have the acetyl-CoA. Okay, right, so basically we're now going to skip a bunch of steps, and we're going to say acetyl-CoA can be used to construct this molecule called HMG-CoA. Okay, so out of acetyl-CoA, you can build HMG-CoA through a bunch of steps that we are just going to abbreviate down to a line, basically. Okay, so what does HMG-CoA stand for, firstly? Okay, well, the full name for HMG-CoA is beta-hydroxy. Okay, so the first H there is for the hydroxy. Then it's beta-methyl. Okay, that's the M, so HM. And then finally, G is for glutarol. So beta-hydroxy, beta-methyl, glutarol, and then it's coenzyme A, which I'll just abbreviate down to CoA. Okay, so that there, which I'll underline in orange, is the full name for HMG-CoA, beta-hydroxy, beta-methyl, glutarol, coenzyme A. Okay, so what then is the structure for this? Okay, well, to understand the structure of this, you firstly need to know what glutaric acid is. Okay, so glutaric acid is a 5-carbon molecule where at each end of the carbon molecule you're going to have carboxylic acid groups. So let's start by drawing out that. Okay, so here is one carboxylic acid group, and I won't draw the alcohol group because it's actually going to be linked via a phyroester link to coenzyme A, but we'll add that on in a moment. Okay, so here are the 5 carbons. One of these carboxylic acid groups will actually be free. Okay, now under physiological pH it will have lost its proton off that oxygen, okay, which is why I'm drawing the oxygen with the negative charge rather than the alcohol group. Okay, so here one carboxylic acid group is free. In glutarol-CoA, the other carboxylic acid group will be linked to coenzyme A here. Okay, so once again represent the CoA as a turquoise box here. Okay, so that's the CoA. So glutaric acid then is just a 5-carbon molecule where you have carboxylic acid groups at both ends, and if we were just drawing pure glutaric acid we wouldn't have this CoA on here for a start, but we would have hydrogens then coming off these free carbons and saturating them, so each carbon would have 2 hydrogens. However, I haven't put the hydrogens on yet, and the reason is because obviously we don't just want glutarol-CoA, we want beta-hydroxy, beta-methyl glutarol-CoA, so these portions need to come into consideration. Okay, so the beta, what does the beta mean? Well, this is the old way of naming up the carbons in a carboxylic acid. So in the modern era, the way we name up the carbons in a carboxylic acid is we call this number 1 because this is the more important carboxylic acid group because it's got the coenzyme A attached. We then call this number 2, number 3, number 4, number 5. However, in the old days, the way they used to name up the carbons in a carboxylic acid molecule is they'd call the first carbon off the carboxylic acid group the alpha carbon, then the next one would be the beta carbon. Okay, so that's what this beta-hydroxy, beta-methyl is referring to. This carbon here, This is the beta carbon, basically. Okay, so we're going to have a alcohol group and a methyl group coming off this carbon here. So this carbon will have an alcohol group coming off, that's the beta hydroxy, and then it will have a methyl group coming off it, that's the beta methyl. Okay, and then all we need to do is finish the molecule off by putting hydrogens then off the remaining carbons. Okay, like so. So this then is the structure of beta-hydroxy, beta-methyl, glutaraldehyde-CoA. And once again, I'll stress the fact that this oxygen doesn't have the proton attached simply because under physiological pH, usually that proton will be donated away into free solution, and therefore it would usually exist like this as the conjugate base, basically. Okay, right. So this is the structure of beta-hydroxy, beta-methyl, glutaraldehyde-CoA. So out of acetyl-CoA molecules, specifically out of 3 acetyl-CoA molecules, you can build one of these structures, basically. Okay, now comes the really important reaction that I actually want to look at in detail because it's the rate-limiting step and also because it's the site of action of statins, which are one of the main anti-atherosclerotic drugs that we will eventually come to discuss. Okay, so the next reaction then is the conversion of beta-hydroxy, beta-methyl, glutaraldehyde-CoA or HMG CoA into a molecule that is called mevalonate. Okay, so let me firstly draw out the structure of mevalonate and then we'll discuss what is actually going to occur in this reaction and we'll discuss the enzyme which catalyzes it. Okay, so this portion of the molecule remains completely unchanged. Okay, these 4 carbons with this methyl group coming off the side here, this is all unchanged. It's this portion that's going to be hit by this conversion. So we'll firstly just draw out the unchanged portion. Okay, so this carboxylic acid group has not been affected. It will still have that proton missing under physiological pH, hence why we're calling this mevalonate rather than mevalonic acid. Okay, mevalonate's denoting the conjugate base here. Okay, so then off this beta carbon we'll still have the methyl group and the alcohol group like so. Then we'll have another methylene group like so. And now, this is the portion that's changed. So off this first carbon now, instead of having a carboxylic acid group, we're going to have 2 hydrogen atoms and then an alcohol group. Okay, so basically what has happened is you have reduced the carboxylic acid group to an alcohol group. If you remember back to your chemistry, you can do that. You can turn carboxylic acid groups into alcohol groups. They firstly go into aldehyde groups and then they go into alcohol groups eventually. Okay, right, so that's what we've done basically. We have reduced the carboxylic acid group into an alcohol group and we now have this molecule mevalonate. Okay, right. The other product, of course, of this reaction is that you'll also get the coenzyme A regenerated. So now it has its intact phyl group there. Okay, and it can now go off and be free. Okay, right. So how do you do this reaction? Well, basically, in order to do this reaction, you need a lot of hydrogen atoms to be provided. And where do we get hydrogen atoms from in biology? Okay, well, we usually get them from NAD or NADP that has been reduced. Okay, in this case, we get them from reduced NADP, basically. Okay, so what you're going to actually need in order for this reaction to occur is you're going to need 2 reduced NADP molecules to come in and provide the electrons that they are carrying, okay, along with that proton. Okay, now let me just talk about what NADP molecules actually carry. So NADP molecules, they carry what's called a hydride anion. Okay, so I'll just put this over here. Okay, a hydride anion, and this is basically a hydrogen atom with a negative charge. Okay, it's denoted H negative like so. So what this effectively is it's a proton, okay, at the center of the hydrogen atom with 2 electrons circulating around it, or circling around it. Okay, so here are the 2 electrons here. So they have 1 proton and 2 electrons. Now, the normal unreduced NADP molecule has a positive charge. Therefore, when you combine it with the hydride anion, it then becomes neutral, and that's what's denoted here by NADPH. Okay, so this is the normal NADP without the hydride anion with a positive charge. That positive charge will be neutralized when you add on the hydride anion, and it will become this reduced NADP here. You're going to bring in these 2 NADPH molecules, these 2 reduced NADPs. They're going to return down to being 2 non-reduced NADP molecules, and therefore you're going to provide into this reaction 2 electrons from each of them. Okay, so 2 times 2 electrons, effectively 4 electrons in total, and also 2 protons in total, 1 proton from each of them. Now, in reality, what we need for this reaction is we need 4 protons and 4 electrons. We've got the 4 electrons here, okay, but we need another 2 protons. Where do we get those from? Well, we just get those from free solution, basically. So you need 2 reduced NADP molecules to come in, and you also need 2 protons to come in. Okay, and that means that overall, then, we are now putting in 4 hydrogen atoms, basically, i.e. 1 proton and 1 electron together. That's a hydrogen atom into this reaction. Now, why is that exactly what we need? Well, let's have a look at this. So basically, if we want to convert HMG CoA into mevalonate, let's think about what sort of bonds we need to break. We firstly need to break that bond between the carbon and the sulfur atom there, that thioester link. Okay? Basically, and this is not intended to be an electronic flow diagram, this is just intended to make you understand how what's on this side adds up to what's on this side. Okay? Basically, you can imagine sending 1 electron in this bond back to the carbon atom and 1 back to the sulfur atom. So imagine breaking this bond, sending 1 electron back to the carbon and 1 back to the sulfur atom. Okay, that leaves the carbon and the sulfur atom each with an unpaired electron. Then bind this carbon and this sulfur to a hydrogen. Okay, so that uses up 2 of our hydrogen atoms, basically. So 1 is going to bind to that carbon and 1 is going to bind to that sulfur. That will regenerate us the coenzyme A here. It'll also then give us an aldehyde group. Okay, like so, you'd have a carbon atom double bound to an oxygen, then with a hydrogen here. Now what we want to do is put in the 2 other hydrogen atoms, and what we're going to do now is we're going to break 1 of those bonds between the carbon and the oxygen, not both of them, just 1 of them. Okay, again, imagine giving 1 electron back to the oxygen, 1 back to the carbon. Now the carbon wants to bind to something and the oxygen wants to bind to something. Take our 2 hydrogens here, bind 1 to the carbon and 1 to the oxygen, and then you'll get exactly what we've got here, the alcohol group. Okay, so that's how putting in 4 hydrogens can take us to this alcohol group from this carboxylic acid group with the thioester link there. Okay, right, so this is a really important reaction because it is the rate-limiting step in the synthesis of cholesterol. Okay, and it is catalyzed by a very important enzyme, okay, called HMG for beta-hydroxy beta-methyl-gluteryl-CoA. Okay, so HMG CoA reductase is the enzyme that catalyzes this. Okay, because after all, it is reducing the HMG CoA molecule to a mevalonate molecule. Okay, and that's a really, really important enzyme that's going to be the target for statin drugs, which we'll see later on. Okay, right. Then we will abbreviate a huge number of steps now down. Mevalonate can then be used to create cholesterol. Okay, so that's my overview of cholesterol biosynthesis, and really we've only studied 1 tiny little reaction in this great pathway in any detail, and that is this conversion of HMG CoA into mevalonate, which is then used to produce cholesterol. Okay, and that's because this is the really foundational reaction that you need to know about, because firstly, this is the rate-limiting step, which means that the rate of this determines the rate of cholesterol production by this entire biosynthetic pathway, and also because the statins work on the enzyme which catalyzes this conversion here. Okay, right. So this reaction can occur in peripheral cells and also the liver cells. Okay, and it's catalyzed by this enzyme HMG Coreductase. Now I just want to talk about the HMG Coreductase enzyme in a little bit more detail because it is actually regulated by cholesterol levels within the cell. Okay, so at the moment what I want to talk about is how it can be directly inactivated by cholesterol being too high, how it can actually be triggered to be destroyed by cholesterol being too high. And then in a moment we will go on to the SREBP pathway, which is a pathway whereby when cholesterol is too low, you can set up the synthesis of more HMG Coreductase so that you can synthesize more cholesterol. Okay? Right, so let me talk then about HMG Coreductase in more detail. So the enzyme itself is sitting within the ER membrane. Okay, so it's in the membrane of an intracellular organelle called the ER. Okay, now the ER membrane is again a phospholipid bilayer, which I'll represent like so. Okay, so this here, this is going to represent our ER membrane, the phospholipid bilayer of the ER membrane. And basically the HMG Coreductase is going to be an integral membrane protein sitting within the ER membrane. Okay, right. Now it has 8 membrane-spanning alpha helices, and it is a single protein. That's probably the first thing I should say. It's a single protein, so it's not a multi-enzyme complex. It will have an N-terminus and a C-terminus, and between those it will have 8 membrane-spanning alpha helices. So let me just draw now a picture of the HMG Coreductase. So this side of the membrane here, this is representing the cytosolic side, and this is representing the ER luminal side. Okay, so let me just colour in little bits here. So here's the cytosol, here is the lumen. Okay, right, now the N and C termini of the HMG Coreductase protein are both going to be on the cytosolic side of the membrane. So here's the N terminus. Okay, then you're going to have your first membrane spanning alpha helix, your second membrane spanning alpha helix, your third, your fourth, your fifth, your sixth, your seventh, then your 8th, and then on the cytosolic side you're going to have the catalytic domain. Okay, so this is the portion that's actually capable of catalyzing the conversion of HMG CoA into mevalonate. Okay, so let's just colour the different portions in here. So this is the catalytic domain here in orange, sorry, not in orange, in green. Okay, so this is the catalytic domain. Okay, and the other really important portion to stress about this enzyme is a sterol sensing domain, basically. Okay, now I'll colour this in. So 5 of these membrane-spanning alpha helices that are sitting within the ER membrane form what is known as a sterol sensing domain. Okay, so let's say these 5 here, or rather it is these 5 here. Okay, these are going to form what's called a sterol sensing domain, and this is the portion that responds basically to cholesterol. Okay, so let's think about this in a little bit more detail. So HMG Coreductase, it's this incredibly important enzyme in controlling the rate of biosynthesis of cholesterol within liver cells and within peripheral cells. Okay, if cholesterol is very high within the cell, we do not want to be synthesizing more cholesterol. Okay, so we would quite like cholesterol to somehow inhibit these HMG Coreductase enzymes, and indeed it does. So basically, in the 2nd, 3rd, 4th, 5th, 6th membrane-spanning alpha helices here, these form a sterol sensing domain, a domain that's capable of binding cholesterol. Okay, now, when cholesterol is very high within the cell, cholesterol in the ER membrane will go up. So I'll represent cholesterol like so. These are little cholesterol molecules in the phospholipid bilayer of the ER membrane. Okay, so I'll colour them in red, and I've missed the actual molecules. Okay, I was using the wrong side of the highlighter, so let me recolour those in. Right. Okay, so if cholesterol is too high in the cell, you'll also get a high concentration of cholesterol in the ER membrane. Okay, and what can then happen is these cholesterol molecules can bind to the sterol sensing domain. Okay, and when the sterol sensing domain gets cholesterol bound to it, okay, what happens is this is going to lead to the HMG-CoA reductase enzyme getting degraded, basically, getting destroyed. Now, how does this actually occur? Well, basically, when the HMG-CoA reductase sterol sensing domain has cholesterol bound to it, okay, it changes conformation and it can now associate with another protein. Okay, now I'm going to draw this other protein just as a blob because its more intricate structure is still a little bit controversial. Okay, this protein is what's known as an INSIG protein. Okay, and there are two different types of INSIG protein that can both do the same thing. Okay, there is INSIG1 and there is also INSIG2. Okay, and for our purposes they are both capable of doing exactly the same thing. Okay, so we'll always be talking about INSIG1/2. Okay, and they're both capable of doing the same thing. So here it is in blue. Okay, right. So when cholesterol goes up inside the cell, that's going to cause cholesterol in the ER membrane to go up. It will bind to the sterol sensing domain of our HMG-CoA reductase enzyme. Okay, that will change the conformation of our HMG-CoA reductase enzyme so that it can now bind to the INSIG1 or the INSIG2 protein. Okay, and when the HMG-CoA reductase binds to the INSIG1/2, this leads to the HMG-CoA reductase being destroyed by the ubiquitin proteasome pathway. So the protein will be destroyed, basically. Okay, so when cholesterol is too high within the cell, you switch off synthesis of cholesterol, basically, by destroying your HMG-CoA reductase enzymes, which are in the ER membrane. Okay, so that's the way of shutting down cholesterol biosynthesis when cholesterol in the cell is too high. Okay, what we will talk about in the next video then is the opposite thing. When cholesterol is too low, how does the cell then respond? And the cell is going to respond in two ways. Firstly, it's going to respond by making new HMG-CoA reductase enzymes, okay, to start up biosynthesis of cholesterol again. Okay, and secondly, it's going to respond by putting up on its cell membrane receptors that are capable of binding to LDL particles in the blood, okay, and are capable of then endocytosing the LDL particles, and then once we've endocytosed an LDL particle, we can have the cholesterol basically that's stored within it. Okay, so that's another way of getting cholesterol to cells that are cholesterol depleted. Okay, so in the next video we'll discuss the SREBP pathway, which is a response to too low cholesterol basically within the cell.